As our understanding of biological molecules increased in the 20th century, research discovered that all living organisms share a genetic code in 1956. Francis Crick proposed that DNA is an information storage molecule capable of replicating itself. Further, he proposed that the information that was transmitted had to be read by a manufacturing body within the cell which puts amino acids together in the specific sequence, ultimately synthesizing a protein. This became known as the central dogma of molecular biology. Specifically, DNA serves as a template for the direct synthesis of a messenger RNA-mRNA molecule in a process known as transcription. Secondly, mRNA is read at a ribosome by transfer RNAs, tRNAs, which work together to assemble a specific chain of amino acids which collectively assemble to generate a protein in a process known as translation. Proteins are the cell's internal machinery, similar to parts of a car. Each protein has a specific three-dimensional shape that determines its function. Any change in the shape potentially changes the function of the protein. Consider an analogy of transcription and translation to printing an essay from a computer. Once your essay is complete, you store the document to the hard drive. Similar to DNA, the hard drive stores information. DNA is a genetic storage molecule. Data in a computer is stored in a binary language. When it is time to print your essay, you send a command to the computer to send a message to the printer. This message is akin to mRNA, a genetic messaging molecule, similar to the binary message sent to the printer. DNA and RNA share a semitic language based on nucleotides. Hence why the information exchange from DNA to RNA is called transcription and exchange of information in the same language. Once the information is received at the printer, it is translated from binary language into a different language, a language of ink. Analogously, mRNA is read by the ribosome and translated into a language of protein, which are made up of amino acids. Thus, the process from RNA to protein is known as translation, translating from the language of nucleotides to amino acids. The ribosome is akin to the printer, serving as a facility for the process of translation. The molecules that actually translate the mRNA at the ribosome are a different kind RNA, transfer RNA, or tRNA. In the process of translation, a tRNA reads the mRNA, links a specific amino acid to a growing protein. For your essay to represent your idea, the ink must be physically arranged in a specific manner. Any malfunction in the positioning of letters would not convey the same idea. Similarly, proteins have a specific three-dimensional shape that determines their function. Any change in that shape can potentially alter its function. In our analogy, DNA is the stored file in the hard drive. mRNA is message sent to the printer. The printer is the ribosome, the essay is the protein, and the letters represent amino acids. DNA indirectly codes for protein. DNA directly creates all of the intermediate layers of transcription and translation. DNA's day-to-day function is the production of RNA molecules. mRNA is directly generated by a specific segment of DNA. That segment of DNA is known as a gene. The mRNA travels to a ribosome, which is made up of protein and another type of RNA, ribosomal RNA. At the ribosome, the mRNA serves as a code for the synthesis of protein by linking specific amino acid is an exact sequence. The overall collection of an amino acid chain is a protein. DNA is also capable of self-replication, necessary for the creation of the new cells.